Hi, everybody. This is Rick Gordon. I'm Vice President here at Halo Labs, and I'll be moderating today's webinar. Thanks for everyone for joining us today, and I'm really excited to be able to reveal the Aura to you all, because as you're going to see, this is really a game changer in the field of particle and aggregation analysis. Before we begin, I'd like to remind everyone of the question area in your control panel. It should be on the right side of your screen. To submit a question during the presentation, simply type it in the question section. And at the end of the event, we will take some time to try to answer as many questions as we can. If, however, we don't get to your question, we can just follow up with you individually later this week. Or you can email us your questions and we can take things offline. So with that, it's my pleasure to introduce you all to Bernardo Cordovez, and he's the founder and chief science officer of Halo Labs. Bernardo completed his master's and PhD degrees at Cornell University in mechanical engineering, and he has extensive experience in microfluidics, nanophotonics, and particle capture, detection, and analysis techniques. Bernardo has won several awards for his innovations and has been named one of the top entrepreneurs in his field. I know Bernardo is very excited about our newest product, the Aura, so I'll hand things over to him and let him tell you all about the Aura. So thank you, Bernardo.
Thank you so much, Rick. I really appreciate the kind intro, and everybody, really appreciate everybody attending. It means a lot to us, and I hope everybody is well and safe. Like Rick just mentioned, we're incredibly excited to be launching the Aura, and it's a testament of the hard work that Halo Labs has put in changing the field of subvisible particle analysis, particularly in these very tricky times. And my objective with this sneak preview that you guys are getting with the Aura is to make you realize that you're not going to need any other subvisible particle instrument. It is that much better than everything else that's out there, and we've made it our prerogative to address the key gaps in the field. So the main objective of our product line is for you to know what your particles are. And by that, we mean 100% of your particles. We want you to know what they are, how big they are, what they look like, how many of them you have. And we're adding now essentially color to this analysis and enable you to understand which kinds of particles you have. Right now, you have the unfortunate choice in essentially picking between technologies that don't get you there. The Aura addresses all of those gaps. And one key motivation by which we built the Aura is that we interviewed several of you. And one of the most important questions that comes up in protein therapeutic particle analysis is your guys are asking yourselves, is this a protein particle or not, right? And it's a very important question because it marks a fork on the road by which you have to address this problem from a stability standpoint, right? Is it a choice of excipients, or is it intrinsic or extrinsic particles, right? And that's very important for you to know. And unfortunately, right now, you cannot do that. And I'm going to show you why. Expanding on what we've heard from you, we've gotten a number of responses of why protein particle ID is so important. So the first one is that it informs the changes in the formulation buffer. The second one is earlier in the process, most of the particles and the elements that you have there should be proteinaceous. So if something else is going on, that is dramatically important. One of the top 10 pharmas, they give me a really good example that they were optimizing their formulation for protein stability, potentially adding excipients and polysorbate, when basically a year later, they found out that those were not protein aggregates. It was actually degraded polysorbate particles. And because they looked just like protein aggregates, they were optimizing the wrong thing. And they spent a lot of time going into a rabbit hole. And again, they didn't have the tools that allowed them to make the right decisions at the beginning of the process. And the whole idea is for you to create a formulation that has the least number of particulates, proteinaceous and not proteinaceous. This is an example about why this is very difficult. So this great paper from the Japanese Pharmacopoeia came out a couple of years ago, analyzing the fairly new NIST protein mimic standard, ethylene tetrafluoroethylene, ETFE. And I have a small Pepsi challenge for you. For these 3 sets of images, 2 of these images are protein particles. I'm going to give you a few seconds to think which of these 2 sets of images of these 3 are proteins. And if you guess these 2, you have much better eyesight and intuition. I probably would have thought that the non-protein would have been the one at the bottom, but it turns out that the top left image is ethylene tetrafluoroethylene. So the bottom line is that for these pictures acquired with flow imaging, you know, morphological parameters and intensity filters are not enough, and they can be deceiving. And that's one of the key limitations of just doing flow imaging and pretend that it can do particle identification. So what do you have left on the complete tail end of the process, right? So you got spectroscopy, whether it's FTIR or Raman. And what you're seeing here is a Raman spectrum. And it may be confusing, but this is actually a really good result, believe it or not. And you're going to get something that looks like this after extensive signal processing and backgrounding. And you have to be a protein spectroscopy expert to know that, hey, I'm actually looking for this guy, the Mi2 bond. So as you might imagine, that is a pretty painstaking process, right? It requires a tremendous amount of expertise, a tremendous amount of signal processing. You have to manually find those particles. And honestly, that takes almost an infinite amount of time, which is why these tools are only used forensically, right? So basically, you can't do analysis in the early end with the flow imaging, and the only way that you can access this information is at the tail end once you've already failed, and that's simply unacceptable. And the reason for that is that flow imaging morphological parameters are just not enough to do particle identification. You have low signal to noise images due to the low refractive index contrast. And with Raman, for example, it's also a low signal to noise signal. There's weak nonlinear scattering signals. And they're both time consuming. Flow imaging is much more time consuming than BMI, and Raman is dramatically time consuming. So you're only working with measurements of n equal to 1 at a time. And they have very well known volume limitations, and they're very much they require extensive amount of manual work, as I'll describe later on as well. So to fill this gap, we created the Aura because we want you to find the particles that you need to characterize at the very beginning of the process so that you can make good decisions from the beginning. And we do that by not only improving background and membrane imaging, but by also bringing about fluorescence membrane microscopy. And that's one of the key focuses of this presentation. And this is a representative image of a protein and a non-protein mixture in our new setup. And the red particles are proteinaceous and the blue particles are not proteinaceous. So we're adding color to your particle species so that you can actually tell them apart. And I'll dive on this in much more detail as the presentation goes on. But you can actually give color to these black and white images that characterize flow imaging and even BMI. So we are enabling you to get a much more deeper understanding of what's actually going on in your sample. So this dramatic improvement over the state of the art, it enables you to do protein, non-protein ID, to count your particles. It has 100% sampling efficiency, and I'll show you why. And by that, I mean that you count every single particle that's there. You can get quantitative results down to 5 microliters of volume. We can measure very small particles to very large ones. It's very fast, 1 minute per sample. It's high throughput. It's compatible with robots. And on top of that, it has high refractive index contrast measurements built in, which is very important to characterize your proteinaceous populations. So how does Aura enable high throughput protein ID? So the offering of this product line is threefold. So we have the instrument, which enables high throughput bright field and fluorescence particle detection. You have new consumable plates that support fluorescent workflows. And one key feature is we have intuitive and flexible analysis software, which we're very proud of and allow you to characterize every single particle with a tremendous amount of detail. And you're going to be able to get knowledge from the single particle level to the sample level at the well level to high level insights to protein on protein. So it's like 4 tiers of incredibly important information that the system is going to allow you to get in very little time. So let's start with the improvements over background and membrane imaging. So one of the first ones is that we now have 100% sampling efficiency. Most of you, I'm going to go quickly over this. Most of you are familiar with how BMI works. It's very simple. First, you background the plate. You put the plate into the Aura, or in your case, the Horizon instrument, and you take an image of the background. You then load your sample and filter it, and then you put it back in the instrument to measure the filter rate where the analysis happens. Expanding on that, you take a picture of the background where you see the morphology and the topography of the background membrane. You then pipette your sample onto the membrane. You then place that sample in our vacuum manifold, and you suction the samples through the membrane. And you put it back in the system, and the image that the system is going to acquire, this measure image, is going to be essentially the convolution of the membrane and the particle images. But because you already have the background image, we can do a pixel by pixel subtraction using great software that allows you to see particles that would have been very difficult for you to do with just a manual method. So the software is key in revealing these high-level insights and being able to tap into the smaller particle size populations and even measure the larger ones with much higher fidelity. So the tiers of BMI analysis, as you well know, is we take pictures of particles. So you have particle level information, their morphology, their size. One tier up, you have particle size distributions, which is the well level information, how many small ones and how many big ones you have. And then higher level information is this aggregate analysis, so that you truly understand what's happening with your entire particle population in your experiment, but powered with amazing data visualization techniques. And we basically improve on that significantly as well. And again, one of the key enablers of background and membrane imaging is that refractive index really matters. And we have 4x or more higher refractive index contrast than whether it's flow imaging or light obscuration. And that is a key enabling technological advantage that allows us to tap into these very transparent particles that are protein aggregates, and even AAV aggregates for that matter. So I told you something that you already know. So what is new with BMI? We now have 100% sampling efficiency, and we have data analysis that makes it easier and far more powerful than ever before for you to get to both deep level and high level insights. So let's get started with 100% sampling efficiency. So first, 100% matters. If you look at the Horizon instrument, image is about 66% of the membrane, and then we have an exclusion area that you effectively analyze 63% of the sample. With Aura, it has amazing new optics that allow you to sample the entirety of the well. So that means you get to count all of your sample. You do not need to apply any scaling factors. There's no data acquisition knobs compared to, for example, flow imaging, where you set your frame rate or your flow rate, and little to no sample optimization is required for you to measure 100% of your samples. So that's one key advantage right here. And I'm going to do a little bit of a parallel and talk about flow imaging. So if you guys are familiar, that typical flow cell height is going to range anywhere from, say, 150 micrometers to 300 microns, depending on the objective that you're using. But basic optics, right? If we just go to the Nikon Microscopy University links, you get to see that a 4x objective with a 0.1 NA, the depth of focus is 55 microns, right? I mean, this is how a 4x 0.1 NA objective works. So that means that you're sampling you're properly sampling 55 microns instead of 300 microns, right? So a lot of the flow imagers, they've chose to change the conversation of sampling efficiency into how many images you can take in a sequence. But it doesn't address the basic fact that only a fraction of your flow cell is properly imaged. So not that many of them are in focus. That's just by design. And even if you had a 100 micron channel and use a 4x objective, that's only going to be 50% that are probably going to be in focus. Then you have to deal with the fact that these are not consumables, that these are flow cells that you have to do a very diligent job cleaning. And this paper that analyzed the impact of the dirt on flow cells for both light obscurations and flow imagers found that there's an exponential impact, particularly at the smaller size ranges, right? That, you know, fouled flow cells can count 10 times more in the lower ranges than the non-fouled flow cells. So you actually have to worry about this. It's not just a routine, but they actually impact your measurements. So that's essentially unacceptable the way that we see it. So put together a accurate list of, say, here's 13 reasons why we're better than flow imaging, why BMI coupled with FMM is better than flow imaging. All of your particles are in focus. We don't have to set the frame rate and the flow speed. Both of those are very manual and critical to the output of these data. We have high refractive index contrast. We are not a consumable, so you do not need to worry about washing or fouling or the impact of the consumable walls, scattering light. Our throughput is 10 times faster, if not more. Our volume requirements on the lower end, we have 2 orders of magnitude lower volume requirements, which is critical for earlier stage work. The characteristic dimension of these channels, in the case of flow imaging, is 300 microns. So that dictates what is the largest particle that you can properly measure. In our case, our characteristic dimension is between 5.6 millimeters, which is the diameter of the well. And then in the case of light obscuration and flow imaging, your sample just ended up in the drain, so you cannot ask any more questions. While in BMI and FMM, you can ask a lot more questions, either by doing subsequent, for example, washes to figure out what are the soluble elements in this formulation, to actually do fluorescent staining, as I'll expand on as we move forward. And with fluorescence membrane microscopy, you can actually ID protein particles as well. So there's basically no reason why you shouldn't just be using the aura. So with BMI, you get accurate sizing and large counts dynamic range. So you're 15, here we're showing how 15, 10, and 5 micrometer beads measure, dead on their corresponding size. But we've improved the optics. So now we can measure particles smaller than 2 microns. And here is a highly concentrated linear dilution of 1.6 micron beads. And we are linear over 3 million particles per mil for particles smaller than 2 microns, right? So the linearity enabled now by the better optics of the system is dramatically improved over the existing Horizon instrument. And also very importantly is we've now shown that you can obtain linearity at ultra low volumes. So you can get quantitative results for down to 5 microliters or even 10 microliters per well. And here's linear dilution data for 5 microliter wells containing human IgG protein particles and 10 microliter wells of human IgG particles as well. And you can see that the data, the R-squared values are basically 1, and the linearity is basically outstanding. So you can get truly quantitative data for very, very small volumes. And that's very important, again, for earlier stage work. So as I mentioned before, we're working with black plates as well as these new plates that support fluorescence workflows, as well as white plates. Some of you may be wondering, well, how do they actually compare? You can see that they look a little bit different, but at the core, they're essentially offer the same performance. Let's start with some images of ETFE. The top panel shows an ETFE image gallery acquired on a white plate, and the bottom panel shows an ETFE image gallery acquired on black plates. So you can see that you still have these really nice contrast images. The ETFE looks, for lack of a better word, black with a nice gray background contrast. So essentially the images look the same. And more quantitatively, when we compare the counts between the white and the black plates, here we have rotated human IgG aggregates in a citrate buffer for both plate types. And because we're pipetting in different sets of plates, right? One white plate for the left bar and then a black plate on the right bar, you can see that essentially you're getting counts, within the error of the pipetting. So they compare, the images look the same, and the counts are basically the same as well for this particular sample. So we talked about how we improved background and membrane imaging, and most of you are probably wondering, okay, so let's talk more about protein on protein ID. How does Aura enable you to do that? And we do that via labeled fluorescence. So to summarize the key advantages that we have over the other technologies quickly is that we are much higher throughput, we have much higher signal to noise, we have 100% sampling efficiency, low expertise, low volume, and we can do particle ID. There's the throughput of Raman is dramatically low. FTIR is a little bit faster than Raman, but it's still, you're working at single particles at a time. The signal to noise for Raman is just unfortunately dramatically low by design. It's very specific, but dramatically low. And most of these systems, certainly Raman and FTIR, require a lot of expertise to use, the flow imaging a little bit less, but you still need to know which knobs to click if you're going to get good data. And all of the techniques above are essentially volume limited if you don't run them properly. So let's talk about our workflow. So how is this different than BMI? And the short of it is that it's not that different from BMI, from the simple, easy to use BMI workflow at all. And we have 5 easy steps to follow for one of our recommended workflows. And basically steps 1, 2, and 3 are BMI. You background the plate, you pipette and filter your sample, and then you measure in bright field. That is exactly how BMI works. But then we add 2 very easy steps, is then you take the plate out, you put it back into vacuum manifold, and you pipette 40 microliters of fluorescent dye. You don't really even have to wait. You suction that sample, and then you put it back in the system and you measure the fluorescence. And one key advantage from that is, as you can see in steps 1 through 3, we're doing BMI, right? So we're getting the right particle population counts just like you would today. So we're essentially decoupling any effects of staining onto the measured particle mask, right? So we apply the fluorescence after the fact, and it results in some very nice data. That's called membrane phase staining, and the membrane phase staining kinetics are very fast, so it makes you not want to wait, as I'll show on in a couple of slides down. But the system is very flexible. So most of you have in the label fluorescence work that you've done before using plate readers and spectrophotometers and whatnot. Most labeling is typically done in solution, and this system supports that as well. And in that case, you would background the plate, you would mix your formulation with the dye of interest, and then you would load this mixed sample, right? Your sample plus the dye, and then you measure in bright field and fluorescence. So that is supported as well. And I think one of the beauty of this approach is that you don't have to choose, because the software is smart enough that it knows how to align the fluorescence images with your bright field images. So even if you wanted to use a hybrid setup in which you mixed membrane phase staining with solution phase staining, you can. So let's dig in on this. And one good example, I think I'm going to take you back to this Japanese pharmacopoeia sample. So how can Aura help you distinguish the undistinguishable, right? So I don't know if you guys remember, but I think I even forgot which of these is the ETFE particles. I believe they're the ones in the top left. How can we use this fluorescence labeling to differentiate proteinaceous versus non-proteinaceous samples using the Aura? And we're going to use this first workflow which again is steps 1 through 3. We're going to conduct background and membrane imaging to measure your particle distributions. And then after that's been done, you now have your sample filter on the membrane, bring it back to the hood, put it in a vacuum manifold, and apply 40 microliters of 5 millimolar thioflavin T, as I'll describe, and then we're going to measure it in fluorescence. So here's the setup, right? For this experiment, we're running a controlled experiment in which we're running 24 wells of rotated human IgG. We're running another 24 wells of this NIST protein mimic standard, which is reference material 8634. And then in column 7 through 9, we're running 24 wells of serial mixes of these two materials. So first we pipetted and filtered the ETFE, and then we pipetted IgG over it so that we know how much ETFE and IgG is present in these serial mixes, and it allows us to then verify these results not only in counting mode, but in fluorescence mode later, plus the appropriate negative controls. So to do this, we're actually going to use the gold standard for protein aggregate staining, which is thioflavin T. Many of you may be familiar with thioflavin T. It's probably the most popular histochemical dye to stain amyloid fibrils in vitro, and it's quite commonly used for neurodegenerative diseases such as Alzheimer's and Parkinson's. These amyloid fibrils are characterized by cross beta sheets as the primary second structure, and there's a lot of literature on thioflavin T since it's been used for a while. Some of the mechanisms of interaction are still being disputed. One of the ones that's most well characterized is that it essentially binds to some of these pockets in these fibril beta sheets. We like using thioflavin T because it's very well established. It's even used in other instruments as well. And it's highly stable and water soluble. So that means you can use a lot of it. And the fluorescent characteristics, excitation and emission, are also very well characterized. And our first fluorescent channel is designed to work with thioflavin T. And here's what the molecule looks like. As I mentioned before, the first workflow supports membrane phase staining. The image on the left shows after we've already measured the bright field images, we're going to pipette 40 microliters of THT, suction it, and then measure it immediately in the system. And you can see that those red particles are saturated signals coming off of these human IgG particles. And the image on the right is the same thing after 3 minutes. So we are exploring unique membrane phase staining kinetics that you really don't need to wait and sit around. The results for not waiting for the sample to incubate over the membrane versus waiting for several minutes, it doesn't make much of an impact, and it allows you to use the tool routinely. So you don't have to worry about overnight incubation or any of that. Back to the experiment that I showed you before. Again, it's a controlled experiment. So we have our ETFE particle counts, our IgG particle counts, and these serial mixes. And the reason we ran this controlled experiment is you can see that we have about 50,000 particles of ETFE. We have about 140,000 particles of IgG, and my IgG plus ETFE serial mix is very much within error of the sum of the ETFE plus IgG, right? So that's a good way to cross-reference the results once we apply our fluorescence analysis. This is now what the new image set looks like. So the water so we applied water and then we filtered water and then we applied 5 millimolar THT over that water control. You can see that it looks pitch black, right? So so the membrane essentially supports the fluorescence and supports any background fluorescence inherently from this thioflavin T. But if you look at the human IgG aggregates, they fluoresce very strongly, and they show these, in this case, we show these blended images of fluorescence in which the fluorescence is captured in red. In the bottom right, these blue particles are actually ETFE. The fluorescence image is blank, and we're actually using a different filter, the SIMI filter, our side excitation light scattering, to be able to pick up these particles and even color the images, the particles according to their ETFE signatures. And in the bottom left, we have a mixture of the two, where you can see the red human IgG aggregates and the blue ETFE. So we have not only the chemical separation, but even unique light excitation sources that allow us to distinguish particles even more in the case of SIMI. So let's look at a typical plot. So if you have rotated human IgG and you plotted fluorescent intensity in the Y versus diameter in the X, this is a typical scatter plot that you're going to see for a given well. And what you can see is that fluorescence signal is increasing, with particle size. It makes sense. There's more pockets into which this TAT can bind to. But I think the most important characteristic is that it's significantly above the black background. So these are 8-bit images. And what that means is that, say, 0 is black and 255 means white. It means a very bright particle. So you can see that there's a distribution far above 0 for human IgG, meaning that it's stained strongly. If you look at the same case, however, for the ETFE, you can see how the fluorescence intensity for the ETFE is dramatically low, and most of these particles are far below 20, right? So there's a gigantic difference in the fluorescence properties, even though optically ETFE and proteins, as you saw, they can basically look identical. If we now look at fluorescence versus diameter for this mixed particle population, you can actually start to make out the two particle populations with the proteins with fluorescence intensity significantly above 20 and the ETFE essentially not fluorescing, being very dim. So they do not fluoresce essentially because they're not protein aggregates. And there's many ways in which you can analyze this. I'm going to show you 2 ways in which how we can separate out and tease out protein versus non-protein for this mixed sample. So what we can now plot many wells at the same time, and we're going to plot fluorescence versus diameter. You can now collapse all of this well data and see it all in one shot. And you can manually say, okay, I'm going to set a threshold, and then you get a horizontal and a vertical bar, and it's going to split the analysis into quadrants. So the horizontal bar was picked at around 2 fluorescent intensities, and the vertical bar at around 5. And I'm going to show you that again, and then we're going to lock the thresholds. Just to make sure that that's captured, let's do it again. I selected, say, 6 wells. We can now plot fluorescence or even relative fluorescence, which is scaled from 0 to 100, which is nice for grouping data. All of that data is now collapsed in a single graph. And I can now split the analysis for that group data into quadrants. And that's very, very nice. And we can do this manual setting thresholds analysis. And basically, the software is going to split up this data set into 4 quadrants. Q1 is in the top left, Q2 is in the top right, Q3 is in the bottom left, and Q4 is in the bottom right. And it's going to tell you how many particles are present in each quadrant. And through that analysis, we now have one of the advantages of this system, both BMI and FMM, is that it does a really nice job grouping data. You can see that for the top right quadrant, which in this case is Q2, not to get confused with Q1 if you're trying to go, counterclockwise. But the top right quadrant shows you that for the ETFE samples, only 244 particles per mil of your almost 16,000 particles per mil for the greater than 5 microns fluoresce above the background. So you basically have almost no ETFE. 1.5% of the ETFE cross this threshold, and they could easily be other things present in the ETFE solution. So the ETFE does not fluoresce, and we can get that quantitatively. Now, if we're looking for particles greater than 5 microns, we have about 4,300 human IgG aggregates, and you can see that those that fluoresce are basically almost 100%. 99.8% of the IgG fluoresced very strongly and is captured in this quadrant. In the case of the serial mix, that number is 88%, meaning 88% of the particles are above this, are inside this quadrant, and 12% are outside of it. And I'll explain that in more detail. If you go back to how we set up, so this experiment was run in a controlled fashion, right? So we know how much we were putting in there to begin with. That's why we ran a serial mix. So if you just use counts, right, in this controlled experiment to tell you how much ETFE is present in the serial mix, you're going to see that only that 15.5%, you would calculate that 15.5% of the serial mix had a ETFE component for the greater than 2 micrometer particle population, and for greater than 5, that's 14. If I do a fluorescence-based approach, I basically get, which has no idea how these serial mixes work, you get basically identical results. So we're able to replicate the counts, and the counting is accurate for splitting this protein and non-protein population via this fluorescent thresholding analysis. And you can do it manually. One great way of doing this as well is with one of the most powerful features of the software, which is called the expression engine. So as you well know, the software stores all of the information for these particles, whether all the morphological parameters, the sizing, the semi-intensity, the bright field intensity, and now even the fluorescent intensity. And this expression engine allows you to basically use Boolean logic, and it sounds harder than it is, but it's actually super easy, to tell you how many particles meet my criteria of interest. So we have these preset expressions that you can use to tease out, for example, which particles fluoresce above the background. It looks harder than it is, but basically what this expression is saying is FL1 intensity is the average intensity for a given particle, and it's basically tells you count it if the fluorescence intensity is greater than the background plus 6 standard deviations above the background, right? So you're giving yourself a confidence interval, and you can report it for particles greater than 2 microns, 5 microns, etc. It's a really powerful way of getting high-level insights, and certainly I use it all the time, and it's actually very easy to use, and it exploits the fact that we have all of this information there and we don't want to let it go to waste. But there's more to just separating protein and non-protein. As you remember, the bottom half of the fluorescent particles were ETFE and the top was IgG. What about the middle? So we're going to click this particle somewhere in between. You look at it in black and white, you don't know what it is. If you look at side scattering, you're going to be able to see that that if it scatters strongly in the left, the fluorescence on the right, and if I look at a combined image, you can see that it's non-protein on the left and fluorescent on the right. So that's a mix, right? So let's look at this as well. So again, the ETFE does not fluoresce. The populations at the bottom, the IgG populations scatter strongly. Let's look at the inflorescence. If I look at the semi, it clearly has edges on the left-hand side, but it has strong THT fluorescence on the right, and we can see that that one's actually a mix. So you can do much more than high-level insights. You can really dig deep. And because we have, in addition to fluorescence, many more morphological parameters, you can analyze these samples in meaningful ways that was simply impossible before. For example, we are now plotting fluorescence in the y-axis and semi in the x-axis, right? And you can tease out these two very different particle populations. So you can see that there's a population that essentially has no edges, that fluoresces very, very strongly, and another particle population that has very well-defined edges that does not scatter at all. So this is one of the very powerful features of the software, is that in addition to the fluorescence, you have all sorts of different parameters that you can use to separate out the particle populations. So some of you may be wondering, okay, that sounds interesting. What about some very important negative controls? So a common one is polysorbate. We generated palmitic acid particles by degrading it. And this degraded polysorbate, it did not fluoresce into THT. I hope none of you are storing your formulations inside Corning cryovials, because those shed particles, they look just like drug product particles. But because they're not protein particles, they also do not fluoresce in our first channel of THD. And here's an example of a stainless steel particle. I mean, that's more for downstream processing, but stainless steel is not protein, so it does not fluoresce. Another very important particle of interest for many of you is silicon oil. So we formulated a unrealistically concentrated silicon oil sample. It's a 2% silicon oil, which is basically over 100x more than you would find in a pre-filled syringe. And we have a bright field image on the left and a fluorescence image on the right. And what you can see is you can almost see these smudges, right? Because we just have such highly concentrated silicon oil that you can actually see it on the membrane. But when you stain this well with THD and you look at it in fluorescence, these smudges are not staining, and you can really see in fluorescence the human IgG particles fluoresce. So you don't have to worry about not picking up your protein content, even at unrealistically high silicon oil concentrations. And the software is more powerful than ever, right? So you can see the entire full well image, you can see it in fluorescence, you can do blended images in which you zoom in and you see your protein particles. In this case, I'm looking at the ETFE signature through SIMI. You can then go to the scatter plot. You can plot all sorts of different parameters. We're going to plot it fluorescence versus size again. That's a common setup. We can size it by any parameter of interest. In this case, we're going to visualize it by circling by the dots by size. And then we're going to collapse this data into a single data set, right? And you can basically pick 4 or 5 or however many particles you want to see, and then look at the data individually by zooming into each one of these images individually, or look at the aggregate result. This is an example of selecting 32 wells. You can honestly visualize up to 96 wells if you wanted to, and then collapse all of that data such that you get to understand, you can see essentially every single particle in your experiment and see how does that particle relate to the rest of your population. Again, at the individual well level or at the collapse level, which is very powerful. But wait, there's more. Once you're working with fluorescence, it opens up a lot of very important avenues, and we have the option of adding a second channel. And one of the very common applications is to, in this particular space, is to look for lipid particles such as polysorbate or even oils as well. So we basically provide more information than any other tool. I did a lot of explaining of the protein on protein capability. We can look for lipophilic and hydrophilic particles with hydrophobic stains. We can get an understanding of the 3D morphology of the particles with the SIMI intensity. And this is a nice image on the right that shows palmitic acid particles mixed with human IgG particles, and essentially this 3-channel blended image in which you can see all of these different elements at the same time. So it's an incredibly powerful tool. The fluorescence, the second fluorescence channel can basically be anything you want. We do have some standard configurations for staining lipophilic particles. TMA DPH is a common one that is supported. Many of you may be familiar with BODIPI. We support BODIPI 630, 650, which is meant to stain lipophilic particles and even hydrophobic proteins. And a paper from an important pharma CRO came out using dye stain for staining hydrophobic particles. So all of those are supported, but basically you can pick virtually any excitation or emission channel for your needs. Here's an example of palmitic acid particles being stained with TMA DPH, and they fluoresce very, very strongly, and that is available. So in conclusion, why Aura? I have to tell you the truth, why not Aura? I have no idea what you would use anything else. But we basically enable high throughput particle analysis. It now takes a few hours instead of a few weeks. And no washing. We enable high throughput particle ID. If you compare it to Raman, I'm saying a few hours, really it takes 1.5 hours to run 96 wells and get the protein on protein ID, as opposed to a few months if you really wanted to characterize all your particles with Raman. We're quantitative down to 5 microliters. We measure 100% of your samples. We get high level insights and detailed insights of the sizing and morphology. It's very fast. The software is amazing and flexible. The images come in high contrast, which is very important to characterize protein samples in AAV and a lot of the biologic samples. And it comes with 21 CFR Part 11 software is available as well. So thank you so much for attending. Again, we're super excited about this product and I hope you enjoy it.
Thank you very much, Bernardo. That was a great presentation, and hopefully everyone got some good information out of it. So as a reminder, on the right side of your screen, there is a section where you can ask questions, and those will come in, and I'll try to get, there's a bunch of questions that have come in, and I've tried to answer some of them as the presentation has been going on. There's still quite a few questions out there that we need to address. And so let's go ahead and get started. And the first question that we have is, can the second channel also be used for other protein dyes?
Yeah, that's a good question. And the short answer is yes. It can support dyes, for example, the Alexa dyes, which are meant to bind to monomer. One interesting capability of the system is that unlike, for example, flow cytometry or flow imaging when used in fluorescence, is that it extracts out all of the monomer content, right? So you would be able to use monomer dye staining. You drain out all of the monomers, and you can use Alexa dyes to actually stain the protein elements of your protein aggregates. So that is unique. Compared to what these other systems offers. So the short answer is yes.
Great, thank you. The next question was, how much extra time does the fluorescence measurement add to the workflow?
So again, BMI takes about an hour, right, for backrunning and measuring 96 wells. The fluorescence step is it's just like doing like another backrunning or another measurement step. So it's about a half hour for 96 wells. So I think it's about 30 to 35 minutes for the 96 wells.
Okay, great. So this question, we got a couple questions, I think, around the fluorescence workflow and how to kind of understand that a little bit better. So this question, I think, might answer several of them, which is, how do you ensure the fluorescent dye particles are not confused with stained protein particles?
So if you may recall, I presented 2 workflows, and I used the workflow in which you first run BMI, right? That is, you background the membrane, you pipette and filter your sample, and then you measure the bright field, right? Which that's essentially BMI. At that point, you've properly captured there's no dye involved at this step, right? And you've already characterized your particle size distribution. Then you take that plate for which you already have your particle size distribution completely untampered. You put it back into manifold and you pipette 40 microliters of THT dye. And you conduct membrane phase staining, right? Which, again, I mentioned before that you don't even have to wait very long at all after you pipette. After that, you suction and dry that plate, much like you would, again, in a regular horizon workflow. You bring it back into the Aura, and then you click, you load the tray, and then you click run the fluorescence measurement. and the system will automatically align the fluorescence images with the bright field images. So again, your sample has already been measured. The system doesn't know in advance, right, that you have dye. But what that really allows you to do is that it's not perturbed, right? So your particle distributions are unperturbed, and you're only looking at, of those particle distributions that I already characterized, of those particles that I already measured, how many of those fluoresce and how many don't. So that's one of the really powerful advantages. And if your dye, for example, happen to have any external contaminants, or it, for lack of a better word, it's not properly formulated, or or you let it sit for a long time and you start seeing some precipitation, right? Those dye particles would probably fluoresce very strongly. But the good news is that you don't have to worry about that because you already know where your real particles are. So you're only looking at the existing particle mask, at the existing particle population, and within those bounds, do they fluoresce or not? So you don't have to worry about these external contaminants, and you don't have to worry about perturbing the sample, for example, through incubation in advance.
Okay, great. Thank you, Bernardo. This one was one person asked a question about expression engines, and kind of two related questions. Can any measured parameter be used to search or characterize things with the expression engine?
Yes, so the software stores all of the morphological information. So you've got size, circularity, minimum and maximum and mean ferrets, semi-intensity, bright field intensity, average fluorescence intensity in channel 1, and even average fluorescence intensity in channel 2. So yeah, you can, every single one of those is stored and can be used, and it's actually quite simple. You can just say, is, for example, the average fluorescence intensity greater than 30? And it'll just pop out, here's how many particles met that criteria. And you can even build much more complicated, Boolean logic by adding, for example, and or statements. Like you can say, and the diameter is greater than 10, and semi is greater than 5, right? And then you can just tease out very nuanced differences to accurately identify your particles or the particle population of interest. But while that may sound complicated, we have pre-built and, for lack of a better word, canned expressions that are used, for example, to separate whether particles fluoresce significantly more than the background already to begin with.
Okay, great. Two questions related to volume. The first one is, what volume of dye is needed to sufficiently label the particles? And I guess that's probably with the first dye.
40 microliters of 5 millimolar THD is what we use. And that's a good volume for fully encompassing the well. Many of you here might already be using 40 microliter samples, for example, to load your formulation. So it's very similar, and it's just 40 microliters does the trick.
Okay, great. The second volume related question was, you mentioned small volume a couple of times in the presentation. Can I run larger volumes if I want to?
Yes, there's a couple of different ways you can do that. Either by running multiple wells is probably one of the easiest ways to get started. Some have done continuous filtration over the same well as well. But yeah, you can run larger volumes as well in a couple of different ways, whether you run multiple wells or whether you continue to pipette on the same well. We typically recommend running more than one well for that.
Okay, great. And then there were, I'll probably answer this question. There was a couple of questions related to, I think there must be people who currently have a Horizon system. The questions were, can my current Horizon be upgraded to an Aura? We don't change out parts in the Horizon system, so we don't swap out parts of the Horizon system. We do have a trade-in program, so if you do have a Horizon, we can trade that in towards credit. The optics and the internal components of the Aura are quite a bit different than the Horizon, so there's no real upgrade path. It's got a very different camera. It's got a lot of internal guts. However, if you do have an Aura, you can always add second channels later. And so you can get it with one channel of fluorescence, and then you can always upgrade to a second channel of fluorescence. So that's relatively easy to do. by our technicians. The other one, there was a couple questions that people had about robotic compatibility and what sort of liquid handlers. So we do have many customers who, even with the Horizon, they're having their plates be pipetted by liquid handlers. So like TCAN, Hamilton liquid handlers can certainly grab these plates, dispense liquid onto these plates, and manipulate the plates. The change with the Aura is now the Aura, it has all the software hooks to talk to a liquid handler. It also has a fully robotic compatible plate tray, so that the robot hands can, the grippers can get in there and put a plate into the Aura. And so you can have the Aura system essentially working for your liquid handler and completely automating your workflow. So that's a new thing. Okay, great. And then the other last question I think we'll probably have time for, we're running out of time here, is sampling efficiency. So you mentioned 100% sampling efficiency in Aura. How does that compare with things like flow imagers?
Yes, so flow imagers have chose to basically redefine that conversation, right, about what sampling efficiency really mean. So the way they define sampling efficiency is because they're taking several pictures, right, as these particles move in these microfluidic channels. They're basically trying to, they define sampling efficiency as the overlap or the lack or the gaps between pictures taken, right? So in my opinion is that that's just disingenuous, because it's not taking into account the Z, the depth discussion. And as I presented before, if you have a flow cell that is 300 microns and you use, or even let's call it 100 microns, right, which is a fairly thin flow cell, and you use the lowest magnification objective, right, and just the optics are such that if you use a 4x.1 NA, you only have 60 microns of depth of focus. Only 60 microns of those 100 microns are going to be in focus. And it's well documented that particles are missed in FlowCam. So not only are they missed, but they're literally essentially out of focus. So yeah, so if you look at their literature, they're going to say 85%, 90%. But that is basically just redefining the conversation of how many pictures in a sequence I can take, and it's not looking at the Z depth. If you take that into account, if you look at a 300 micron flow cell, which is recommended for the 4x objective, you do that math, they're not using sheath flow or anything. Yeah, only 20% of those 300 microns are going to be in focus. So I would say, yeah, it's somewhere between 10, 20, and if you use a thinner channel, at best you're going to get 50% of properly imaged particles. And properly imaged, properly detected in focus should be how we define sampling efficiency, and now basically steer the conversations of how many pictures I can take in a row.
Yep. Okay, great. So we're about out of time. There's still a bunch more questions, and so what we'll do is we'll send these questions out via email to you individually. So we have your name that's associated with the question. I did also just want to thank everybody again for attending today's webinar. Thank you, Bernardo, for presenting. This webinar will be available on our website, so it'll be recorded. It is recorded, and it'll be available on our website, in about 24 hours. You should receive a link as well. Great, thank you, everybody.